I'd like to introduce Dr. Kurt Brunden. Kurt is the Director of Drug Discovery and Research Professor in the Center of Neurodegenerative Disease Research within the Perelman School of Medicine at the University of Pennsylvania, where he oversees drug discovery and translational research programs in the areas of Alzheimer's, frontotemporal lobar degeneration, and Parkinson's disease. And we are discovering, by the way, that the dementias are related more than we had ever believed beforehand. Dr. Brunden's work has been funded by NIH grants and through a number of collaborative research programs with pharmaceutical and biotechnology companies. Prior to joining CNDR in 2007, Dr. Brunden was an executive in the Biotechnology Center, where he served as Vice President of Research in Gliatech Incorporated and later as Senior Vice President of Drug Discovery at Athersys Incorporated. In these positions, he initiated and managed drug discovery and development programs in Alzheimer's disease, dementia, schizophrenia, inflammation, metabolic disease, and cancer. Prior to joining industry, Dr. Brunden was an NIH-funded faculty member within the biochemistry department at the University of Mississippi Medical Center, with a research focus on the regulation of myelation. He obtained his Bachelor of Science degree, magna cum laude, from Western Michigan University with dual majors in biology and health chemistry, and his PhD in biochemistry from Purdue University with a postdoctoral fellowship at the Mayo Clinic. Dr. Brunden has more than 125 scientific publications, 11 issued U.S. patents, and foreign equivalents, and several pending patent applications. Dr. Brunden received a $50,000 CART grant in 2010. And by the way, Dr. Brunden, I was there to see you present in 2010 and 2011. Thank you for that. I still don't understand what you said, but hopefully you'll clarify that. He got a follow-on grant in 2011 for $250,000. He is the only researcher that has received 2 CART grants. He was a colleague of the late Dr. John Trojanowski, who served on the CART Scientific Review Panel for almost 20 years. And with that, welcome, Dr. Brunden, and thank you for everything that you have done for us and for research.
Well, thank you very much for the invitation to present, and thank you very much for certainly the funding that CART has provided me in the past. And what I hope to do today is to give you an update, if you will, on some of the work that the CART did fund back in the, I guess, almost a decade ago, or over a decade ago now, and hopefully show you that, you know, we've made some pretty significant progress. And in fact, I think we have what could be a potential drug candidate ready to start going into the studies required to get into human clinical trials. And hopefully, Bill, this time you'll be able to understand what I'm saying. And if you don't, please, everyone, I welcome you to, you know, ask questions along the way if there's something you don't understand, because there's nothing more frustrating, I know, than to sit for a half hour and having lost the thread in the first 5 minutes, if you will. So let me share my screen. And can you see those slides? Can anyone see those?
Yes.
Okay, good. I'm going to get rid of this little thing. I guess it's okay. Okay, let me give you the full size here. Okay, so today I'm going to talk about a program that, again, was started a little bit before the CART funding, but then really took off, you know, in the subsequent decade afterwards with, you several NIH grants that have led us to where we are. And it's really about the development of a class of compounds that target something called microtubules. And so they're called microtubule stabilizing agents. And we'll get to what microtubules are in a moment. And so, this is kind of the perfunctory introductory slide that I'm sure all of you have seen at one point or another, some variation. Obviously, this group's well aware of Alzheimer's disease and the prevalence and the unmet medical need, although there is a little bit of hope on the horizon, as I'm sure you guys are aware of, with the new A-beta antibodies. Since I've been working in Alzheimer's now since the 1990s, the vast majority of drug development programs, both within industry and then even in academics, have focused on one of the two key pathologies in Alzheimer's, these amyloid plaques. And here's a nice picture, showing what a plaque might look like in the brain at a magnified level. And these plaques, as you may or may not know, are extracellular deposits of this misfolded peptide known as amyloid beta. And there's clear evidence from genetics and inherited cases of Alzheimer's that, mutations in the protein called the amyloid precursor protein, from which this peptide, the amyloid peptide, is derived that forms these plaques, that they can cause inherited disease. So plaques are clearly important in Alzheimer's disease. But there's a second pathology called neurofibrillary tangles or tau tangles, and those are found within cells and within the nerve cells, as depicted here. And it's both of these pathologies that are really required to cause Alzheimer's disease. And in fact, even today, diagnostically, a definitive Alzheimer's diagnosis is typically made, an autopsy, looking for the presence of both plaques and tangles. Sorry, let me go back a slide. Now, in addition to these key pathologies, of course, there's other things like glial cells, which are support cells in the brain, like microglia and astrocytes, that can become kind of inflamed, if you will, in Alzheimer's disease. And there's clearly evidence to suggest that they're maybe contributing to the disease process. And ultimately, of course, it's the loss of these neurons that are involved in the learning and memory and everything else that goes on in your brain, essentially from a cognitive standpoint, that leads to the symptomology of the disease. So, when I entered Penn in about 2007, as I said, a lot of groups were focused on the amyloid plaques and trying to find ways to diminish the impact of them. And there'd been very little work on the tau tangles. And over the subsequent decade or so, there's been a lot of work done on understanding the disease progression. And a lot of studies have benefited from the identification of new biomarkers that allowed us to kind of look at changes through either imaging or through changes in proteins in the cerebral spinal fluid that you can tap from the spinal cord that kind of represents the fluid of the brain, looking for changes in markers of disease. And, I'm not going to go into all the large set of data that were used to generate a graph like this, but what we've learned is that changes in the amyloid peptide that make up those plaques occur 10 to 20 years before symptoms in Alzheimer's. And in fact, you can image these plaques by positron emission tomography, so a brain scan, if you will, and they've pretty much hit their plateau well before symptom onset in Alzheimer's disease. In contrast, the tau protein that forms these neurofibrillary tangles in neurons can now also be imaged, and the development of these tangles increases during the disease phase. And so unlike amyloid, where changes occur a decade or two before disease symptoms, tau changes during the disease. And in fact, there's a very high correlation between the amount of tau pathology burden in your brain and your cognitive symptoms. And that's kind of depicted here. This is now a positron emission tomography ligand that can be used to scan for tau in brain imaging studies. And the more color you see, that means the more tau pathology. And if you map across patients with high pathology have lower mental scores. Okay, this is a mini mental score exam where a high number like this is pretty much normal, and then as you go to lower numbers, you're worse off. And so there's a very high correlation between the formation and progression of tau pathology in the brain and the cognitive symptoms of the disease. And that really leads us, and now many others, to think that tau is ultimately the killer, if you will, of the neurons in the brain. Amyloid beta peptide and the plaques are clearly important, but they happen way earlier, and they're probably involved in ways we still don't fully understand in helping drive the tau pathology. So that has led us, in my group, to largely focus on tau and how might we intervene to affect, the tau pathology in the disease. Not ignoring the amyloid plaques, but, putting a little emphasis on the tau. And so what is tau? Tau is actually abundant protein in neurons, and it normally associates with structures called microtubules. And so here's a little cartoon of a neuron. And, neurons have these long projections called axons. And, axons are essentially the wiring of the brain, where they ultimately end in what are called synapse and can, trigger release of synaptic transmission and to the next neuron in line, if you will. And that's, the whole process of brain function and learning and memory, et cetera. And the microtubules serve as essentially the railroad tracks along these long axons. And so they are involved in shuttling things from the cell body way up here down the length of the axon and then shuttling things back up towards the cell body. And so you can really think of these as the equivalent of railroad tracks. And tau binds these microtubules and helps provide stability and function to these microtubules. So that's what tau normally does. In Alzheimer's disease, changes occur in the brain, perhaps driven by the A beta plaques that we talked about, that lead to tau to become modified in a way through a process called phosphorylation. There's a chemical change. It doesn't really matter from a detail perspective that that. It's just that changes occur in the tau that promote its dissociation from these railroad tracks, these microtubules. And that frees up tau then to form these neurofibrillary tangles that you see in Alzheimer's disease. Now, as we were thinking about tau and Alzheimer's disease, clearly these tangles probably aren't good, but the other consequence of tau dissociated from microtubules and forming these tangles is the microtubules are no longer fully stabilized, if you will, by the tau protein. And so that might suggest that there could be microtubule dysfunction in Alzheimer's disease and other diseases where there's tau pathology, like frontotemporal dementias. And, if the microtubule isn't working right, if the railroad track's not working right, then the train can't get up and down the track, right? And so it could be cellular dysfunction as a result of this microtubule not working well. And so that led us to ask, way back in about 2009, 2010, when about the time we got the first CART grant, to ask the question, might there be a way to compensate for this tau loss of function that's no longer bound to the microtubule with a molecule that might bind to the microtubule and essentially serve the role that tau would normally do to help stabilize the microtubule. Okay. And, this idea that was presented on the past slide isn't just fantasy, as far as microtubule structure being altered in Alzheimer's. If you look at Alzheimer's brain, there's clearly a reduction of stable microtubules. It's been reported, been looking at, sections from human brain. The number and length of microtubules and axons is decreased in Alzheimer's disease. And in mouse models of Alzheimer's disease that we and others have, where they form these tangle pathology, you also see changes in microtubules and axonal function and transport. So there's data out there to suggest that this microtubule abnormality or dysfunction is in fact occurring when tau is no longer bound properly to the microtubules. And so again, to use the railroad track and railroad car analogy, if you think of the railroad tracks being the microtubules, and there are literally microtubule motors that act like the engines that move things up and down in axons. If the tau is no longer binding and the tracks are no longer stable, what you end up with is axonal dysfunction. Okay, you're not moving things in the neuron properly back and forth. In essence, a train wreck within the brain. Okay. Any questions so far? Is this clear to everyone? Okay, I'll take a absence of questions, hopefully mean that you understood. And so what do we do about this? Well, interestingly, there are known drugs that do bind to microtubules, and they're used in cancer. And so you may have heard of something called Taxol, for example, or Taxotere. These are cancer drugs. Actually, they're natural products. Taxol is derived from a tree bark, a natural product, that binds to microtubules. And in rapidly dividing cells like cancer cells, they overstabilize the microtubules in a way that prevents cancer cell division. Okay, at high doses, these drugs can prevent cancer cell division, and that's why they are anti-cancer agents. Now, what we asked back in around 2009, 2010 is, what if we took a low dose of something like a Taxol at lower levels than would needed to block normal cell division? Might you still be able to have a positive effect on the microtubules in the axons that are compromised in Alzheimer's disease? It was kind of a long shot, right? We're asking to find this low dose that's going to be beneficial in the brain without having side effects in the periphery. Now, we were initially stymied because the drugs that were available, like Taxol, et cetera, don't get in the brain. Fact is, 98% of drugs in the world don't get in the brain because of this thing called the blood-brain barrier that is meant to protect the brain from things that are floating around in the bloodstream. But ultimately, we did a large survey of other microtubule stabilizing agents that had been described, and we found a class of compounds known as the apothalones, which again are natural products. They've been used, for treating cancer a little bit in clinical trials, but hadn't progressed to approval like the taxanes had. And we found one in particular called apothalone D, which I'm going to call EPO-D for short here, that showed good brain penetration. And we showed it could actually stabilize microtubules in the brain through assays I'm not going to describe here today. And so this led us back in 2011, 2012, after doing a variety of studies to convince ourselves that this EPO-D really did what we hoped it would do in the brain as far as microtubule stabilization. We went in with this molecule into a transgenic mouse model that forms the neurofibrillary tangle pathology reminiscent of what you see in Alzheimer's disease. And that's kind of depicted over here. Let me change to a laser pointer. It's easier to see. And so these are brain slices. So these are half a brain, a hemisphere from mouse, this transgenic mice that form this tau pathology. And as we go this direction, we're going from young to old. And as they get older, they start forming pathology, which is stained brown here by an antibody that recognizes misfolded tau species, the tangle species. And these are just different brain levels, just showing as with the age, they get more and more of this pathology, okay? And what we did is we took these transgenic mice known as PS19 mice, and we started treating them with this EPO-D at about 9 months of age when they have small to moderate amount of tau pathology in this zone. And then we aged them for 3 months, giving them the drug or not, just the vehicle alone, and ask where they would normally get to about this phase of pathology. And we asked, could we see benefit in these mice that had been treated with this drug for 3 months? And so we looked at a number of endpoints. We were looking at microtubules, of course, because that was the target we were trying to hit, but then axon health, axon function, because that's, as I mentioned, the microtubules are the railroad tracks. If we can correct them, hopefully that improves axons. We did behavioral testing. These mice, as they get old, show cognitive deficits, just like Alzheimer's patients. They show loss of neurons, just like Alzheimer's patients. Okay, so we looked at brain pathologies. But the other key thing we're interested in is safety. Because again, remember, these drugs at high doses have been used for cancer, drugs like this. And at high doses in cancer patients, you will see some side effects. because the drugs stop dividing cells, they can stop blood cell division, for example. And so patients get like anemias and things like that because their blood cells aren't dividing properly. They can also get what are known as peripheral neuropathies, where the peripheral nerves get too much microtubule stabilization, and that can be detrimental. And so we were looking for this kind of sweet spot of a dose that didn't cause any side effects, but might have benefit. And the doses we use actually were about 30 to 100-fold less than this drug had been used in cancer clinical trials. So we were quite low in dose, 0.3 and 1 milligrams per kilogram once a week. And long story short, and this is all work now that's a decade old, is we found great benefit by treating these transgenic mice that form tau pathology with this microtubule stabilizing compound, pothylone D. If we do a cross section of a nerve that's in the brain area, the optic nerve that ultimately connects to the eye, we can see in these mice clogged up axons. And so these are essentially, each of these are axons, these structures here that have this myelin wrapping around them. And there are many disfigured ones. Here's one that has a bunch of stuff clogged up. This one's clogged up. And we could do, through analyses of these types of images, blinded, where the person doing the analysis didn't know the treatment type, analysis of how many of these, blocked up dystrophic axons we could find. And what we found is that compared to wild type, non-transgenic mice of the same age, there is an increase as expected in these tau transgenic mice. And when we treated with a drug at both doses, but a little bit of a dose effect, we saw a normalization. We reduced these dystrophic blocked up axons, okay, because we were increasing normal axonal transport, movement of things up and down the axon. We also increased the number of microtubules. These animals show a deficit that when treated with drug actually will increase microtubule density, some maybe even a little more than what they started with. And so we were doing what we expected. We were affecting microtubules. And importantly, we blocked neuron death. And so here is a picture of the hippocampus. It's a structure in the brain that's involved in learning and memory. And all the brown here are the neuron cell bodies in the hippocampus. This is a normal wild-type mouse at about 12 months of age. Here is this tau transgenic mouse at 12 months of age. And hopefully you can see that the hippocampus is smaller here. The number of brown cells is much reduced compared to here. And so what we did was analyze how many neurons within a fixed area, as depicted by this rectangle, were there in transgenic mice compared to the wild-type mice, and did the treatment with the pothylone D, the microtubule stabilizing compound, lead to reduced loss of neurons? And that's what we saw. So again, here's a wild-type animal. The transgenic animals have decreased neurons in the hippocampus, and when we treated with the pothylone at the highest dose, we essentially saved all the neurons. So this was pretty dramatic data. We also showed improvements in cognitive performance, and I'm not showing you today, less synapse loss. That's the point where the neurons connect to each other. It was a quite positive result. So positive, in fact, that, I'm sorry, I got one more slide here. The other thing that we saw in these animals that was somewhat unexpected is we had a reduction in the actual amount of tau pathology in these mice. Now, remember, we were trying to compensate for the loss of tau function at the microtubule. We weren't necessarily trying to, undo the neurofibrillary tangles that were going to form, but in fact, the compound did. So wild-type animals had no tau pathology. Here, the brown staining is the tau tangles and the tau, what's called neuropil threads, the tau that goes into the dendrites of the cell. And when we treated with this pothylone D, you can see there's a lot less of this brown staining. There's still some, but it's a lot less, okay? This is using an antibody that recognizes the highly phosphorylated tau, which is what you see in disease. This is the misfolded tau. It's the same story. The compound reduced the amount of tau pathology. We sort of think we understand why now. It turned out the amount of tau pathology is affected by axonal transport. So by correcting axonal transport, we reduce tau pathology, but it doesn't really matter. The good news is how it does it. The good news is we not only reduce, axonal dystrophy, we save neurons, but we also reduce the amount of pathology. Okay. And all this happened without any side effects. We looked at blood cell counts. We looked at peripheral nerve function. No negative consequences of treating the drug at this lower dose. So all that ultimately led to, computer's a little frozen here. There we go. Sorry, I jumped too far there. There we go. Led to a pharmaceutical company to look at this compound in Alzheimer's patients. So this apotholone D, was actually owned from an intellectual property perspective by Bristol Myers Squibb, the pharmaceutical company. And they had progressed it, to phase 2 clinical testing in humans for cancer, but then put it on the shelf because it didn't look any better than Taxol or other drugs. We were using it as researchers, kind of agnostic as to who owned the IP rights. We just wanted to see if this approach of stabilizing microtubules would work. They saw our data. They ran some, repeat studies to convince themselves that what we saw was real. They replicated our data, and they ended up in around 2013, 2014, taking apotholone D back off the shelf and ran a small study in Alzheimer's patients called a phase 1B study. So this is really only in about 40 patients, mostly looking at safety. They used doses based on the low doses that we had used in our mouse studies. And they just looked at safety, but they did look at some biomarkers in the cerebral spinal fluid, as I mentioned, and they did look at cognitive performance. But this study was only 9 weeks in duration. Keep in mind, most Alzheimer's clinical trials are about 2 years in duration. So, you don't see enough decline in 9 weeks in an Alzheimer's patient to see an improvement with the drug. So, why they looked at this, I guess, just for completeness, but no one would expect a change there. The good news is there were no safety issues with the drug. Of course, they didn't see a change in cognitive performance or biomarkers in that short 9-week study, but again, not surprising. The bad news was this, again, was around 2014. Bristol Myers Squibb, as an organization, made the tactical decision to exit Alzheimer's research because at that time there had been so many different failures of Alzheimer's therapeutics that they thought it was just too risky. And so this compound never progressed beyond this one trial. So this now is about the time that we started talking with CART and others. And we said, well, we need to find new molecules and forge ahead without Bristol Myers Squibb. And so what we did, without, again, going through a lot of legwork that we had to do, is we identified new classes of small molecules that weren't natural products that acted in the same way as apotholone D. And the CART funding, again, helped us in our initial search for compounds that might serve as EPO-D did, but for which we could now perhaps take them forward and progress them towards the clinic. And to make a long story short, we have now focused in on this class of compounds with the tongue twister name triazolopyrimidines, which I'll just call TPDs. And they act very much like the taxanes and the apotholone D that I just described. And what we did with our first prototype compound, TPD, known as CNDR51657, was to replicate the study I just showed you in the PS19 tau transgenic mice with apotholone D to ask whether our compound could show the same benefit. And these are the same endpoints I just told you about. We looked at axonal dystrophy, and we saw that, again, of course, the PS19s have an increase in that. And our compound now, at 3 or 10 milligrams per kilogram twice weekly, again, reduced that axonal dystrophy, just like apotholone D had done. We normalized microtubule density, just like apotholone D had done. And we decreased tau pathology, just as we've seen before. And so we were getting essentially the same results now with our compound. That was much easier to synthesize, had better, what I'll call pharmaceutical properties, if you will. And importantly, again, it was safely tolerated. We didn't see any changes in organ weights, blood cell counts, etc. So this was published about now 5-6 years ago. And we did some more work, and we started getting interested in this other pathology I talked about, the A beta plaques. And that was because about the time that 2018 paper came out, and paper came out from a group at Northwestern University saying that, you know what, microtubules aren't just messed up where there's tau pathology, they're also messed up where there's plaque pathology. And if you look at a transgenic mouse that forms amyloid plaques, what's been known for quite some time is the plaques here are stained in blue. And it's a little hard to see sometimes because the blue and green are together here. But wherever there's blue, there's typically some of this green. And that green is staining for what's known as amyloid precursor protein, the protein from which the amyloid beta peptide is made that forms the plaques. And the amyloid precursor protein accumulates in these swollen axons around amyloid plaques. And what this group at Northwestern showed is the reason that this stuff accumulates in these swollen axons around plaques is because microtubules get disrupted around plaques. And that can be shown, the microtubules stained in red. If I pull away the other stains, you can see these divots in the areas where you see blue, and blue here is a little masked, but where there's plaques, there are swollen axons where the microtubules have collapsed. So all these holes here are collapsed microtubules. And what that group at Northwestern postulated was what happens, again, here's a crude drawing of a neuron with the cell body, the axon, and when the axon comes into contact with the plaque, for reasons we don't fully understand, the microtubules collapse, things that move down but then just stay here, they accumulate, including things like the amyloid precursor protein. And this group said, well, maybe this accumulation of amyloid precursor protein leads to even more amyloid release and even more plaque formation. And we said, well, maybe. And if that's true, and we treated with our new microtubule stabilizing drug so that things could move back up and maybe get rid of some of this accumulation, maybe we could reduce amyloid plaques, just as we could reduce tau pathology in tau transgenic mice. And so we did a study in what's known as a 5xFAD mouse model, but this is a plaque only. There's no tau pathology here, only the amyloid plaques. And here they're stained in a purple dye. So this is the brain with plaques. When we treated for about 2 months with our prototype microtubule stabilizing compound, our compound now, that's 51657, indeed, we saw a reduction in amyloid plaques. To our knowledge, this is the only class of compound that can affect plaques and tangles independently. And so, obviously hitting the two key pathologies in Alzheimer's. And the reason we think that the plaques are reduced is what we found is we also reduced the accumulation of this amyloid precursor protein in other proteins in these swollen axons. And so we normalize microtubule function, reduce plaques. So in the Alzheimer's brain, a compound like this could reduce both of the key pathologies. So that paper was published just a couple of years ago. And during that period, from about 2019 to now, we've been funded with a very generous grant from the National Institutes of Aging to further improve and develop our small molecule microtubule stabilizing agents that, again, were first identified back with the time of the CART funding. And we've now found an improved example that we call CNDR 51997 that has undergone extensive safety and efficacy testing that we think qualifies it as a potential clinical candidate. And I'm just going to go through this quickly, but what we've shown is that just like the compound, the prototype compound I just showed you, we can reduce amyloid plaques with this new version, this improved second generation compound, at doses as low as 1 milligram per kilogram twice weekly. And we can have all the changes in the tau transgenic mice, including reduced axonal dystrophy, reduced tau pathology, and salvaging of neurons, and also a reduction in glial inflammation with this new compound 51997. We've done a large number of studies with this compound, things that would be required by the FDA to say it's eligible as a drug candidate. And we've shown that doses about 10 times as high as needed, to get these positive changes in disease models are very well tolerated. So we have what's known as a therapeutic index where, we have some safety margin, if you will, for the drug dosing. And so I'm going to close out here and just say that, what we've shown over all these years is that low doses of microtubule stabilizing agents, now is exemplified by this new compound, 51997, can reduce tau pathology and the axonal dystrophy and neuron loss, et cetera, that occurs in Alzheimer's models. But it also can reduce the A-beta plaque associated dystrophic processes and reduce A-beta plaque deposition. And the doses that are required to do this are quite low and well tolerated. And so based on all this, we now actually just got a grant review that looks like it should get funded to allow us to advance this into what are known as the IND enabling studies, so investigational new drug studies, all the tests that the FDA requires of you to pass before you can get into human clinical trials. And so the hope is we can move this compound forward into clinical trials. And so obviously it takes a village, as they say, and there's been a lot of important people working in my group who've worked on all these projects, some of the key ones highlighted in bold here. And in drug discovery, you have people like me who are pharmacologists, biologists, but you also need chemists. And I've been fortunate to work over the last 15 years essentially with Dr. Carlo Ballator, who was at Penn initially, now at UCSD, but we continue to collaborate, and it's his group that is involved in all the chemical synthesis of the molecules that I've been describing. And of course, none of this could happen without funding. We've had multiple grants from NIH and National Institutes of Aging, but as mentioned, certainly some help from CART. And so with that, I thank you for your attention and would love to take any questions.
How is the CNDR 51997? How is it administered?
Yeah, typically in our animal studies, we do what's called intraperitoneal, so it's an injection. But we know from this compound is orally bioavailable, so that means it could be given as a pill. And so for the studies that we anticipate doing to allow us to get into human studies, there's a prescribed set of studies that the FDA mandates that you do in 2 species, typically rat and dog, for example. We'll be doing those studies with oral drug administration, so pills.
Okay, thank you.
Yep.
I just want to make a comment, Dr. Brunden. I've been watching research presentations for almost 20 years now, and it always seemed like we were looking at attacking the amyloid beta to reduce that, or get the amyloid beta more effectively out of the brain by improving the blood-brain barrier, or looking at the tau tangle misfolding and see if we could stop that. This is the first presentation that I have heard that offers a solution that resolves both issues simultaneously. So I want to say I am impressed, I am excited, I am thrilled. This is the best news I have heard yet. And
well, thank you
for doing it.
I mean, I think we're quite excited, but we're also very cognizant of the challenges ahead, and the one challenge with this class of molecules is safety, because again, things like Taxols and things that have been used in cancer do have side effects. Now, and in cancer patients, you're only administering the drug for a relatively short period of time, be it, weeks to months. Here we would be thinking about, years potentially of treatment. And so what's going to be important for us, we think we can do it based on what we've seen in mouse studies so far, but what's going to be important is to be able to demonstrate that there's a big enough difference between the dose needed to have the desired effect in the brain and the dose that starts showing side effects. You want to have what's called this therapeutic index or safety margin. And so that's going to be the key. I think it's achievable, and that's what we're going to try to do.
Let me ask you a question because, 60 Minutes had a thing on January 14th about targeted ultrasound opening the brain blood barrier. Is that something you could use with your new compound?
The good news for our compounds, we don't need to. It gets into the brain perfectly.
Okay.
So usually certain small molecules like ours, if designed properly, can get into the brain. There's a certain set of parameters that have to be optimized. I won't bore you with, but we get the same exposure in the brain as we get in the blood. Now, for things like antibodies, so the new drugs, Leqembi, otherwise lecanemab, the A-beta plaque targeted antibody, those don't get in the brain well. Only about 0.1% of the administered dose gets into the brain. And so in that case, what you're mentioning, things that might disrupt the blood-brain barrier could be beneficial in getting greater exposure of those types of drugs. Those are what are known as biologics. They're bigger.
Let me ask you further, because having watched that, on TV, they showed the PET scans and MRI scans and all that, and it had dissolved the plaque, the amyloid plaques, to quite a bit. But I think the question that's unanswered is, will the brain rewire itself to get around the neurons that are gone?
Yeah, and that's a very good question. I mean, one thing we do know is once a neuron's gone, it's gone. I mean, there's a little bit of an exception to that. There are certain regions of the brain, including the hippocampus, where there is a little bit of what's known as neurogenesis, new neuron formation. But in general, particularly the cortex, you don't get a lot of that happening. But to your point, it could be that there could be some rewiring. So example, in Parkinson's disease, there's loss of neurons in what's known as substantia nigra that causes ultimately the motor symptoms of Parkinson's. You don't start seeing symptoms in Parkinson's patients until they've lost about 60-70% of those substantia nigra neurons. And that's probably because the other neurons are compensating, right?
Okay.
And so I think, and if you could stop the neuron loss in Alzheimer's, even a patient who had, let's say, a little bit of neuron loss, I think the brain could start to compensate for that, right? But it's always a matter of degree. If you lose too many, I think you make it to a point where you can't. So as is most cases in Alzheimer's, the earlier the treatment, the better.
Where does this compound stand as far as getting FDA approval?
Well, so the next set of studies we have to do, and as I mentioned, I think hopefully, knock on wood, we'll get a new grant to do this. There's about $5 million worth of studies that have to be done to, demonstrate that it passes all the FDA mandated animal tests in rodents and dogs. And so you got to do, show that it's safe at, excess of what you think you need. You got to show that it's not inhibiting cardiac channels and causing side effects. You got to make sure it's not a genotoxic compound. We've done some of these studies and we think we're okay, but they have to be done more formally by the FDA mandated rules. And so there's this whole set of studies that have to be done that's going to take about 2 to 3 years to complete. You got to scale up the synthesis of the compound to provide enough to do all these studies. So the long and short of it is we wouldn't be in a position to start clinical trials probably for about 3 years. But if we can get there, then it should be good. Bill?
I was just going to say, you've got 6.6 million patients in the United States alone waiting for the results of this study. I've got a quick question. Any possibility that this may be effective in fighting ALS?
Well, we haven't looked, but I will say there is some evidence of axonal transport problems in ALS that could relate to microtubule loss. I can tell you a study that we just got published in a pretty prominent science journal a couple months ago that suggests that compounds of this type might be useful for traumatic brain injury and CTE. And you've probably those of you who like to watch football and things may have heard about this CTE. And so we've shown in a mouse model of repetitive mild brain injury that sort of mimics what we think happens in, contact sports, et cetera, that there is axonal, degeneration there, and that our compound actually was beneficial in this model of CTE. So there are other neurodegenerative diseases where this may be useful. ALS, I'm not so sure about, just because we haven't done a lot of studying.
Just one more comment. Dr. Brunden, I understood exactly what you said. That was perfectly presented. And I'll tell you right now, my biggest fear was that there was not enough studies going on the tau tangles, which are the deadliest aspect of Alzheimer's, which actually shrinks the brain. It looked like everything was focused on the amyloid beta. So, man, I got my fingers tightly crossed, and I hope somebody at Smith, Klein, Glasgow, or one of the other major pharmaceuticals is looking over your shoulder to start dumping some money in this to speed up getting it on the shelf, because God knows it's needed. And thank you for that.
Well, we do our best, and even if it's not our compound, if we can just get others to get in this approach and maybe even have a better compound. I don't care if it's me or if it's GlaxoSmithKline, but if someone can maybe use this strategy to the benefit of patients, that'd be great.
The drug, when it was administered via transfusion or in a pill form?
Well, in our animal studies, we did it what's called intraperitoneal. So it's kind of not an infusion, but it's just it's almost like a subcutaneous injection.
Okay.
That's out of convenience, but in patients, we anticipate this will be an oral drug because we know that it can be absorbed orally.
Dr. Brunden, we sincerely thank you for your time. We thank you for your information. This is a terrific presentation. And we're all very enthusiastic about what you're doing.
Great. Well, I appreciate you taking the time, and I appreciate that CART contributions back in the day that kind of got this rolling. And all of you should be proud of the work you do to help in this area. It's the hardest dollar to get, and this is one of John Trochanowski's quotes. The hardest dollar to get is the first dollars, right? Because it's when you're first trying a new idea, it's hard to get NIH funding because you got to generate that preliminary information and data, to convince grant reviewers to fund you. And so that's where, CART grants and foundation grants like that are very valuable. With that, I thank all of you, and keep tuned. Hopefully you'll hear some news down the road here.